A very interesting overview. We will now have a presentation on CERENOVUS, which is part of the Johnson & Johnson family of healthcare medical companies. Now, Dr. Karen Doyle, who is Associate Professor in Physiology in University of Galway, and Dr. Ray McCarthy, who is Senior Research Manager with CERENOVUS, will talk about this company, which has been an absolutely colossal success and good news story for Galway, and is also a very good case for collaboration. You're both most welcome here today. Thank you very much. Thank you very much.
Hi, thank you. Thanks for having me here today. So I'm going to take you through a specific example of how academia, with Karen, the clinicians and industry, CERENOVUS can work together really well. Do you have slides there? Yeah, there we go. So just a little bit about CERENOVUS first. You may know we have origins from Neuravi, which was an Irish startup based in Galway. We were acquired by J&J back in 2017. We're part of Johnson & Johnson, massive healthcare company. And within J&J, we're part of the med device section of the company, which has many arms. Particularly, we're in cardiovascular specialty solutions. CERENOVUS focuses on treatment for neurovascular disease, so stroke, hemorrhagic stroke. In Galway, we're focused on R&D. We started at about 25 people at the time of acquisition, 90 people now, so it's been a great investment by the company. And we've a particular focus on understanding the problem. President mentioned, we start with a problem, then you can find a solution. And we do that under an initiative called the Neurothromboembolic Initiative, and I head that up in the Galway site. And we've several active collaborations, both with the university here and a major one with Karen, and other academic institutions here in Ireland, as well as abroad. Many clinicians here, you know what a stroke is, but there's two main types of stroke: hemorrhagic stroke, where it's a burst blood vessel and you've a leak into the brain of blood, and ischemic stroke, where there's a blockage in the vessel. And really, we're dealing with ischemic stroke in Galway, and the research that Karen is doing is focused on ischemic stroke. It's quite a devastating disease. You've got a blocked vessel, damage to the brain. Time is of the essence, because there's some salvageable tissue that can be saved if you remove this clot quickly. And what we're trying to prevent is this catastrophic death to the brain tissue. So time is really of the essence. And getting this clot out, maybe 30, 25 years ago, there wasn't many options until a drug came around, alteplase, which focuses on lysing the blood clot and dissolving it and restoring blood. Around the early noughties, devices that were introduced through the femoral artery to go up and remove the clot were introduced. They weren't very effective, and interventionists learned a lot over the years and found ways to select patients and better design devices to remove these clots. And around 2014, 2015, a series of trials came out which showed that in fact this interventional therapy can be quite effective if you target the right patients and if you have the right device. And these are the devices we make in CERENOVUS. For the physician, they don't really see the clot in the brain. What they see is something like this, where there should be blood supply. And what they want to do is remove that clot and restore this downstream blood arterial tree to restore flow. Like I said, the physician doesn't see a hell of a lot. This angio on the left here shows a device being pulled out. There's a clot there, as shown here in one of our models. So they don't really see that interaction. And what we're trying to do in our research is, by looking at the clot that's removed, see if we can get a marker, an imaging marker, that could provide some information to the physician. Also, by studying these clots, can we understand the origin of it? In the lab, with our models, we can use, you can't see the bottom of the screen here, but What we can learn from clots that are removed from patients and doing the work that Karen carries out with histological analysis of the clots is recreate these in the lab and see with different composition of clots, we see different behavior in the lab. So on the left-hand side, it's a red blood cell-rich clot. It's been removed back into a catheter with a device. And because it's friable, it can be washed downstream and cause a new stroke or go out of reach of the interventionalists and they can't remove it. Or on the left-hand side, a clot with fewer red blood cells, high fibrin content. This one interacts less with the device. It's hard to engage. And in that instance, you have a different, I guess, performance requirement for the device needed to treat those two very separate types of clot. And in doing that research, we've developed a range of different devices specifically to target those types of clots. One more. Okay, this is my last slide. And this is what the research is about. So until 2015, these clots were either left in the brain or they were dissolved or removed. But now we've had this incredible opportunity. Once these clots can be removed, these, tiny pieces of material that cause such a devastating assault on the brain, now we can analyze them. Until about 2010, there was maybe 100 of these clots analyzed. Through Karen's work and several others, now we're like 1,000, I think, Karen has analyzed. I think 4,000 in the world, and we're learning more and more and more. And that's why we feel it's important to support this type of research that Karen is doing, so that we can better treat the patients interventionally, better recognize the stroke and the intervention in the first place, and have better secondary follow-up for these patients. So, swap over to Karen.
Hand over to me. Thank you, Ray. Hello, I'm Karen Doyle. So just to carry on from what Ray was saying, I thought it might be interesting to first of all tell you about the journey that we've had with the company. So first started interacting with Neuravi as it was back in the day in 2014 when we were looking at reperfusion injury in an animal model of stroke, and at around that time, thrombectomy began to become a very important thing. So we engaged in a project using similar techniques to the ones I used in the previous project, only studying clots this time. And we realized pretty quickly that it was a very worthwhile study, and so it was extended and expanded in 2018. And then my lab was given the opportunity to analyze clots in clinical trial studies with CERENOVUS. And currently, we're just in the process of finalizing a new research agreement for, again, continued collaboration for the next 3 years. So all in all, there's very substantial cash investment by CERENOVUS, and it's a very substantial project. So I just wanted to tell you a little bit about our Restore registry, which is the project that we have been working on. So we collected clots and medical information about the patients, the stroke patients whose clots we have analysed, and with the aim to find out more about these clots that cause the strokes. So we studied the histopathology. We also importantly looked at the heterogeneity per pass, which Ray alluded to, which means essentially the number of attempts that the clinicians make to try to remove the clot. So the ones that take more passes are the more difficult to remove ones. And what is it about those that makes them so difficult? We correlated the composition that we observed with, for example, clinical and procedural information that we were given by our collaborators, and all with an aim to informing improved medical devices design for thrombectomy, and also to discover novel components that may lead to new treatments down the line. So the partnerships involved in this project are my lab, obviously here in Coram in NUI Galway, also CERENOVUS it's a very strong partnership that we have in this space. We've a strong academic collaboration with Mayo Clinic, a lab at Calmaze, and Brzezinski lab in the Mayo Clinic in the US, and also, of course, our hospital partners. So we had hospital partners in Dublin, in Beaumont Hospital, but also hospitals in Gothenburg and in Budapest and in Athens who supplied us with our samples. One of the main things we did was just a histological stain, which enabled us to see the main components. So this is an example of a Marsh's scarlet blue stain, where you see the main things that we see in clots, which are, of course, probably no surprise to anybody in the room: red blood cells, for example, nucleated cells, most predominantly white blood cells, also fibrin, which you can see is kind of the fibrous reddish material that is surrounding little pockets of platelets. And we know that there are other things within that platelet-rich region additionally, which we're very interested in. No two clots are the same. They literally come in all sorts of shapes and sizes, literally one extreme to the other. So all of the samples that we've analysed from the 1,000 cases are shown here, organised with regard to red blood cell composition, which is the yellow, as it appears on Marsh's scarlet blue stain. And as you can see, it's literally one extreme to the other. And of course, the composition of the clot will influence its mechanical properties, which will of course influence how well a thrombectomy approach might actually, how it might cope with the given clot. So just very briefly to give you some of the information that we've discovered. So we do see a difference in the clot composition per pass. So the later passes, you can see pass 4, 5, and 6 there. So when it takes a lot of attempts to remove a clot, they tend to be richer in platelets and also richer in collagen than the easier to remove clots, the ones that come out in the first pass. And of course, you know, information like this is of importance to the medical device industry. It can lead to improvements and advancements. We also looked at etiology, with the effect of etiology on these clot components. And what we observed was there are two main etiologies actually in these large artery clots, the clots that are within these large arteries. And that is ones that are associated with atherosclerosis, so the large artery atherosclerotic type clots, and also clots that arise from the heart, so cardioembolic ones. There are also a lot of clots that it's unknown. It's not obvious what actually has caused the stroke, and these are called cryptogenic often. And what we observed was that the large artery atherosclerotic type clots, they have a different composition to the ones that are cardioembolic. And in fact, the cryptogenic are quite similar to the cardioembolic. So this kind of information can help clinicians to, for example, decide what treatments to put a patient on after their stroke to prevent, for example, a secondary one. We also measured, just got an estimate of the size of the clots, which you see on the right-hand side there. We literally just took an area size of the clots. And what we observed was that the clots of large artery atherosclerotic etiology are double the size of the clots that arise from the heart or that are cryptogenic. So again, very important information for the medical device industry potentially in the development of optimized device design, as is our observation, for example, that there are a lot more fragments that come from the LAA clots additionally. We also took a look, some people who suffer with ischemic strokes are administered thrombolysis, as Ray mentioned earlier, and we looked to see if that treatment had made a difference on the clots that were retrieved. And what we observed was that the actual composition of the clots was unchanged, which is reflected in the pie charts. You can see the same kind of relative distribution of the main components. But the size is different. So thrombolysis reduces the size of the clots without affecting its overall composition. And thrombolysis also reduces the number of fragments. And just to finish up, just to give you an idea of the kinds of impacts that we've had, we've published a considerable number of manuscripts so far on this project. We've developed a very strong international network, and hopefully it's had some positive impacts on the medical device industry with our industrial partners. And it's also had societal impacts. For example, we have trained 10 scientists so far. They're very highly trained in a whole array of different things. And We also have been able to influence the thrombectomy services nationally and in Europe. So, for example, our collaborator in Beaumont Hospital, Dr. John Thornton, is the head of the Irish thrombectomy service, and the other collaborators throughout Europe are very influential in, for example, ESMINT and ESO, which impact on policy with regard to stroke treatment. And also through CORAM, we've had the opportunity to engage in, for example, interaction with the public. We had great opportunity to produce a film called A Tiny Spark through the Science on Screen program. And we engage a lot. We show this to, for example, patient groups in the Irish Heart Foundation in Cree, for example. And I'd just like to finish by thanking people who were involved in the study. So thank you.
Thank you very much. Thank you very much. Dr. Karen Doyle and Dr. Ray McCarthy, thank you very much. Just, if you wouldn't mind staying with me just for a moment, just in case that there are any questions out here for you, because it's a very interesting area. And so we have microphones going around. So if there's any question at all from the audience, just raise your hand and let us know. We can direct them to either of the two. But Ray, I was reading there the other day, you know, you spoke so much about this thrombectomy. I had never heard about this particular fascinating, innovative life-saving treatment that's available. But I was reading, Ray, that only 18% or less of patients have had a thrombectomy, as in mostly, and these people are mostly getting these thrombectomies in Dublin and in Cork. Yeah. So how can we get this life-saving treatment here in the West?
Sure, it is kind of population dependent, so...
Oh, one second now, Ray. If you mind, pull out the microphone there and, yeah, perfect, thank you. It should be on.
You hear me now?
Yeah.
Yeah, so it is a little bit population dependent in terms of the expertise of the physicians treating it. Typically, you want these physicians treating maybe 50 thrombectomies a year. Okay. So, you know, there's various strategies. It's, you know, we have a comprehensive stroke centre that patients are sent to where the expertise is. And you have a good network to get patients to them quickly. Yeah. Now, more and more patients are getting treated, and I think 18% is probably a bit high. I think that's high for Ireland. There's fewer than that, and in the developing world, it's much, much lower. I think actually in Germany, which is the highest usage of thrombectomy, it's at 14%. So the infrastructure is being built. More patients are getting to this treatment, but there's a lot yet to do. I could draw a parallel with MI treatment for maybe 20 years ago. You're just building that network of, first of all, recognizing stroke, going to a centre quickly and getting imaging so you know the extent of the stroke, and then selecting that patient who will benefit from the treatment. So more patients will be treated, I think, in future, and maybe at some point it'll make sense for the Western region to have a comprehensive stroke centre. I don't know if we're at that point yet. It's more a question for the clinicians. So
if somebody in Castlebar gets a stroke?
Yeah, if they get a stroke, I mean, well,
where are they going?
Well, first of all, it's a call to the emergency services. Go to the nearest primary stroke centre where they can get imaged so that they can confirm what type of stroke it is. And, you know, maybe it's an option to take the thrombolysis at that point, or maybe the decision will be made in consultation with neurologist, the interventional team at Beaumont, or one of the other centres who would take them, probably Beaumont, on whether they get the ambulance to Dublin.
As you say, you often hear about people being brought to Beaumont, all right. Karen, how do you, any questions from the audience there on that particular area? No? Karen, maybe just another one just for yourself. Just how do you see the future of collaborations now between the university and the medtech sector?
Oh, I think it's a very fruitful collaboration already, but there are so many diseases that we don't treat fully effectively. There are still plenty of questions out there that we can help to answer. So I think the future is definitely bright for that. Yeah.
Okay, well, listen, thank you. I don't think there's anything, but thank you very much, Karen Doyle and Ray McCarthy. Thank you very much, both of you.